we were having some discussions off camera. Dan, feel free to continue.
Yeah, so I think that we all find that when we're reading about ADCs, there's just a huge amount of information. There's a huge number of different payloads. There's lots of different antigens being targeted. There's different types of linkers. There's different ways of attaching linkers to antibodies. And I think it's easier, rather than getting too lost in all that sea of chemistry and drugs and so on, as to think about what the original problem was. And then you can work your way forward stepwise as to why you need to introduce these new approaches, why you need to introduce new linker types, new attachment sites, and so on and so on. And then it starts to make a bit of sense. And then you can start to see what different companies are wanting to buy and why.
What would you say some of the original problems with the earlier ADCs that were discontinued might have been?
So I think the big problems were homogeneity. So if you're trying to stick your drug onto an antibody, the concept sounds simple, but in practice that's not necessarily straightforward. And what I mean by homogeneity is that if you have a population of antibodies, the number of drug molecules stuck to a single antibody molecule can vary throughout the entire population. And I think in some of the examples I've looked at, there were populations of antibody, some which had a high drug to antibody ratio, and others within that same pool had no drug attached at all. And I think, from my understanding, the techniques to actually separate an antibody that's got a drug attached from an antibody that doesn't probably aren't refined enough, so that you would end up with a pool of antibodies, some of them doing nothing, some of them with lots of drug attached. Then, depending how the drug is attached as well, will tell you how easily that drug's going to come off the antibody, which you want to happen in your target cells, but you don't want to happen in your general system. So if the drug is coming off the antibody and it's circulating in the blood and systemic circulation, it's targeting cells, tissues that aren't supposed to be targeted. And numbers don't look that scary. 0.2% leakage is something I've seen, but that's enough to have fatal, bone marrow issues and hepatotoxicity and so on, so on. So we're talking about really, really effective drugs at killing cells. And it's fine when you take them to the cells that you want to kill, but when they are just released into the system, they're killing everything. So I think that's, to me, the main problem is attaching enough of the drug you want to the antibody and then making sure it doesn't come back off again. And everything else that has evolved from there is trying to solve those two problems in one way or another.
Yeah, because the whole point of it is that it's supposed to be like a heat-seeking missile, more or less. That's what they claim, right? That's the whole thesis of it. But then, if it doesn't do that, one might as well use chemo or a different modality if that doesn't really happen. That it hits the target and then offloads in the right amount at the right time, I guess. And I guess that's one of the things that makes these modalities quite complex, is that you can use a lot of different forms of linkers, you can use a lot of different forms of payloads, and you can attach them in various ways and then get different results, and which target is right for which star ratio is right for which target. It's quite a lot to take into consideration. And that's not even going, okay, should we go dual payload? Maybe we'll go dual payload. And for the linkers, okay, do we go 2x2, 4x4? What linkers do we use? Do we use cleavable? Do we use non-cleavable? It's quite complex.
Yeah, when it comes to deciding on partnering, most of what I've seen is sometimes even a first in class. What's your opinion on that, Kristoffer?
Well, I would say it depends on who the partner is. The first in class might be tricky. I mean, ADCs in themselves are quite tricky, and then to have a first in class for ADC, it would seem like a tall order. to get right. If someone does get it right, it can have huge payoffs, as most of the targets that are well validated are quite crowded. If you look at HER2, if you look at TROP2, B7H3, I think is another one. There's a few targets which are very crowded, and it needs some sort of novelty within those targets, like a new linker, dual payload. Eli Lilly, which we spoke about last week, they recently acquired a dual payload platform from an American biotech. And I know there's a company in Taiwan also working on dual payloads, along with others in China. So it seems tricky enough with ADCs to get them right. But if they do manage to get the first in class right, like they have low tox, they have good efficacy, then it would seem like a slam dunk. But it's not just getting it in the preclinic, because most of them fail in phase 2 or 3, as most drugs do. So it's not that easy to gauge at an earlier stage with some of these, which is why what we've seen is a lot of companies, they would prefer to have some more data even in the preclinical stages. If they do partner, like they have some sort of tox data at the very least. So that's something that remains.
Well, recently we also had a discussion where one of the partners asked specifically for a novel early stage first in class. So and then when we have other discussions, they look for the opposite. So it's quite interesting to see that the risk appetite also varies among most of the large biotechs and the MNCs.
Yeah, absolutely. Although it's usually the bigger ones will usually prefer well-validated targets, and a smaller one, which might have to take a little bit more risk, would seem more likely to partner for a first-in-class as well, since that's it's riskier, but it also has the potential higher payoff than, if you have a new HER2, you would need to beat an HER2, for example, in order to, it has a higher watermark, you would say, in order to get it validated and approved. And that's also some of the things where even in the later stages, there are some ADCs which are good. They're just not good enough, since they are quite costly in comparison to other modalities. So that's also something which is a bit difficult sometimes. They're just not good enough, since there's a lot of competition with other modalities, not just ADCs.
I mean, to take a little bit of a tangent there, when you were talking about clinical trials surrounding these and dual payloads as well, I am curious to know how some of these drugs are going to be measured in a clinical trial. I mean, you're talking about first in class. If we're talking about first in class as a dual payload as well, it will be interesting to know how they're going to decide the contribution clinically of multiple payloads on one drug. And also you have to actually, now that I'm thinking about it, you're adding in a third factor, because the antibody itself can also have an anti-tumor effect. So you've got 3 potential anti-tumor activities working side by side. It'll be, I think in a first-in-class setting, that might be easier actually, but using that in a, as a combination treatment could be very difficult to pinpoint whether your drug's working.
Yeah, well, it will be interesting to see how the field evolves with the various different linker technologies that are being developed, as well as the different payload technologies that are being developed, since, some people have, so that if we take in HER2, for example, some criticism of it might be that, it doesn't work in low HER2 expression for breast cancer, even though some might say that it does to some extent, thanks to its potency overall. But that might be some critique that it's got. And just to mention in HER2, since that is the most successful one in terms of commercial traction so far, and appears to be revolutionizing breast cancer and might even get first in line. I don't know if it is first in line treatment already, but may receive it. Over time. That's pretty interesting.
Yeah, I think the payload's going to be important there as well, because there are some payloads which are, they're actually making new populations of cancer patients because they're so effective. You can actually have like a low HER2 population of patients who were previously untouchable by other therapies. So that is something that I think we're going to see in more and more tumor types, particularly when you introduce bispecifics, trispecific targeting as well, that you can actually take a low population of a certain antigen and actually turn that into a new patient pool and open up your drug to these groups of patients as well, rather than just the traditional high HER2 expressing tumors. And yeah, this is, I mean, it's the next step forward in precision therapy, because I know this is a little bit off a tangent, but from my perspective and from my, the cancer biology I've done in the past, I think the future of cancer treatment is bespoke treatments for all sorts of different tumor types and all sorts of different cancers, and that most cancers will become chronic conditions that you're treated with for life, with an ever-evolving set of drugs and treatments. And I think that's where ADCs have a real advantage, because they're modular. So you can always update the treatment, and you can update the antigen, update the payload throughout a patient's life, potentially, not just thinking about a tumor you're targeting now, but an evolving tumor over time.
Yeah, exactly. It reminds me a little bit of, was the Australian guy that cured his dog that had cancer a little bit, right?
I can't remember the name, but I know the story.
Yeah, so I can't remember it. Basically, it's his dog got cancer. He used LLM to diagnose which type of cancer it was, and used the genome of the dog or the cancer somehow to develop the perfect therapy for that cancer. So it would seem it's almost more personal, and at some future stage, it might not just be we have a HER2 for breast cancer, but we might have something even more specific and more for the individual that is more potent and safe for the patient.
Absolutely.
Hopefully.
Absolutely. And I think that's going to be that's going to be the way it opens up. I think precision cancer therapy, there's a mistake in thinking about cancer as a single disease, but it's not a single disease. It's hundreds and hundreds of very discretely different, changing, constantly evolving diseases. And if you can have a platform that is constantly evolving with that disease and constantly evolving with patient populations, I think that's going to really open things up. I mean, I remember we had seeing a talk from somebody who described some of these treatments a bit like glasses. So if you are someone who needs to wear glasses, when you're not wearing your glasses, you have a sight defect, and when you're wearing your glasses, you're cured in the moment. But take the glasses back off again, you're back to where you were. I think a lot of cancer treatments will follow that kind of model, where you'll be on these drugs for life.
Interesting.
When you're on the drug, you're cured in a sense, but then you come back off a drug, and it'll have to evolve with you.
Yeah.
Seems like an interesting paper.
Yeah, absolutely. Yeah. I mean, without sounding cynical, if you can make a drug that people can live well on for their whole life, and they have to take that drug for the duration of their now long and extended life, that's a good income for your company. That's, you have to be practical about these things. Companies have to make money, and if you can develop these sort of drugs that extend people's lives, give them their quality of life back, and they are on your drug for a long, long period of time, it's one of those situations where everybody wins.
Yeah, I think most people would prefer that than to being on chemo and to have cancer overall. I think they would prefer that most certainly.
Yeah.
And in regards to the payloads, appears to be some new classes of payloads developing. Various different forms. One being the recent deal with Novartis and Myrix, which appears to have validated NMT as a form of payload.
Yeah. So I think this is going to be a completely new approach, and I think we're going to see it far beyond NMT inhibitors as well. We're going to see a lot of drugs that can have some sort of throughout the cell dampening of a certain signaling pathway or signaling mechanism. If you can take that straight to cancer cells, then you can shut down the cells without having to use the traditional approaches that the current payloads use. So the current payloads are typically chemotherapy targeted to the cell type, preventing
like
DNA. Yeah, exactly. These are corrupting your ability to replicate your DNA or for cells to divide, which is how chemotherapies worked in the past, or still continue to work. You target cells that are replicating. You kill all the cells that are replicating. Now you can consider more potent drugs that aren't necessarily targeting replicating cells because you're taking it straight to the tumor site itself. And that's where drugs like NMT inhibitors come in, because these drugs would be devastating systemically, but they can have a massive impact in cancer cells. So they're not just targeting replicating cells in the same sense, but they can shut down whole signaling pathways. I think with the NMT inhibitors, they can shut down aerobic respiration, so the cells can no longer produce energy, and that's the end of them. If your cells can't produce energy, they can't do anything. They can't replicate. They can't continue to divide and grow. And from my understanding as well, there's not much space for cancer cells to mutate around this problem to overcome it. So I think that's where we're going to see the biggest change, that you can now take very potent drugs that you could never have delivered systemically before, but you can start to target them.
Right. Yeah, that's a pretty interesting development to see what will happen with that. And if someone should be interested in NMT payloads, feel free to reach out to us.
Absolutely.
We currently have a mandate for that. But so regarding that, overall on the ADC market, I mean, there's been a lot of hype with ADCs. We see a lot of Chinese developments in ADCs, as well as some European and US-based developments. Where do you see? There's also different DAR, where they have different forms. It's not quite the normal ADC, but they have protein degradation involved with the drug conjugate. No, something like this I've seen in development.
I'm not completely up on this, but from what I can understand from other companies where they have an enzymatic payload, I can imagine then there's a logical step to couple this with something like targeted protein degradation. So if your payload is an E3 ligase that then gets taken into cells and can start, specifically targeting an enzyme an oncogene product that is overactive in a cell, which is as targeted protein degradation works, but you can then maybe feed in a small molecule that would recruit that E3 ligase. I need to look into that more myself, but I could see that being a very logical approach.
Yeah, because it seems like it's not only classical ADCs, but also other modalities being developed along with ADCs, which might be a sort of next generation.
Yeah, I think the thing is that the ADCs just give you that capacity to take whatever you want to a specific population of cells. And, some of the reading I've been doing as well, there's new avenues beyond cancer treatment as well, because you can target immune cell populations or with anti-inflammatory drugs, and I think autoimmune and infectious diseases, immunology. These are going to be where we see further application of this kind of technology. So we now have this capacity to target populations of cells. So I think that's going to open up a huge amount. And also, I mean, I'm kind of making this up in my own head. I haven't necessarily seen this, but with things like in vivo CAR T, if you can have nucleic acids as payloads as well, I could see you targeting populations of cells in a similar method to introduce nucleic acids for genetic engineering, CRISPR, and so on. Again, that's just off the top of my head, but I don't doubt someone smarter than me is doing this.
Yeah. Well, it's yet to be. I'm sure a lot of people are working on similar things already. I've seen a lot of different forms of, a lot of different formats being developed. So it will be interesting to see. But we thought it would be good to touch on this, as it appears to be one of the hottest modalities in cancer at the moment. But we might also see it expand into, as you say, immunology, infectious diseases. Who knows where
obesity.
else?
Yeah.
Obesity. Yeah, could be for sure.
Well,
we saw a company working on that.
Did you not mention before, Ryan, something about adipocytes?
Yes, I did. I'm currently aware of at least 2 companies working on the obesity, but still at the discovery, and a lot of it is undisclosed. So let's keep it like that.
Yeah. No, I think what Ryan's referencing there, the idea then you can target cell populations again for deliberate destruction of those cells. It's sorry.
Yeah, it targets fat cells. You can specifically
nuke the fat cells basically and just shoot them down.
Yeah, exactly.
But it's not the fat outside, right? It's the fat that surrounds your physical organs.
Yeah.
So that's the dangerous fat anyway. Like all the extra fat we have, most of it is not that dangerous, but it's the ones that are blocking the organs, which is dangerous as far as I understand. And there's even some assets in development that are specifically targeting those types of fats that we have seen recently, which
you can also contact us.
Of course,
if you have any questions regarding certain assets, feel free to reach out.
Yes,
absolutely.
Yes.
In particular for payloads, ADCs, and various linkers.
Yeah.
Yes.
But I think, just to touch on as well, I was quite interested with AstraZeneca over the last week, where they have closed off one asset that wasn't achieving what they wanted to see in phase 1, poor efficacy, and a secondary asset that they actually licensed in is doing well in phase 3, and both are targeting the same antigen. And I think this is where you can start to see that payload, linker, and other qualities around the antibody really have a massive influence. So we're quite used to seeing antibodies and describing them as they target this antigen, HER2, whatever, we reference them by their target antigen. But I think that is going to change as we start to see the linker changing, the payloads changing. And what I think AstraZeneca have done recently is something we're going to see a lot of companies do, where they're going to be cycling through all sorts of different combinations of linker, payload, antigen, tumor type, and seeing what works, discarding what doesn't. And I think people who are canny will be able to take advantage of that and watch and see, okay, this is just the natural optimization of a whole platform. And people who are maybe a little bit more easily put off will see, oh, they're cancelling a phase 1 asset. That's a negative signal, whereas in reality, it's going to be part of the process.
Yeah, but they shut it down pretty early as well. I mean, they didn't dose too many patients in the phase 1, from what I gathered.
Yeah.
So it seemed more like they were trying to just save cost. They saw, okay, it's not working as well as we would have hoped, and then you shut it down.
Because they'll have so many similar platforms and similar programs with all the different parts of the antibody and linker and payload that they will just be quickly wanting to push the ones that are working. And if something's not showing immediate efficacy, they will just want to move on.
Well, yeah, we see sometimes a question regarding CMC as well, so that's also something to consider as well.
Yeah.
And then when we talk to the biotech, CMC is always no problem. Then it's no issue.
Of course.
Yeah.
Let's see.
Especially with the dual payload, no problem.
Yeah.
Well, let's see if that holds up under more scrutiny. It's to be seen. But something for the biotechs who are working on ADCs. If you haven't run into this, do think about CMC before getting further along in the development. If you didn't know this already, it might be worth considering at the very least. Manufacturability.
Yeah.
As it's not very. Is it the heterogeneity of the ADCs that's difficult to get right, or what is it more exactly? That's so because it seems like a lot of moving parts that you then have to make fit together. So it seems like a rather difficult process to do this.
Overall. So from what I've seen, this is kind of goes back to what I said at the beginning. Like there's the most straightforward approach would be to stick your drug onto your antibody using some aspect of the antibody that allows something to be stuck to it. Okay, so antibodies are proteins, and proteins are made up of amino acids, and some amino acids are available for things to be stuck to them.
Is it lysine? Is that
lysine, cysteine, yeah. And the original approaches try to stick, well, and some were successful, to stick their drug on directly to lysines and cysteines that already exist in the antibody itself. Problems with that where there's only so many surface exposed lysine residues. Some of your antibody will get covered. Some of your population of antibodies will get covered in your drug. Some of your antibodies will get absolutely no drug. And there was clearly a difficulty in being able to separate antibody that had no drug attached from antibody that had taken up a lot of the drug. So you've got this massively varying DAR ratio across your antibody population. Or targeting cysteines, the chemistry that was used to target cysteines is quite reversible. There's a very well understood chemical reaction that will undo some of these covalent linkage types, and so the drug would come off and then it flows around the system. So what people are doing now is they're saying, right, can we actually put things into the antibody that are going to be preserved attachable sites? So beyond what the antibody already had in its amino acid sequence, can we actually start deciding where the drugs are going to get stuck, how many are going to get stuck on, and things like bioconjugation and genetic code expansion. These are techniques that allow you to put new chemistry into proteins that you can do chemical reactions on, but these chemical reactions don't interfere with biological processes. And this is the sort of strategy that they're going down. And that allows you to then say, right, every single antibody has got 2, 4, 6, 8 molecules attached, or it's got this many linkers attached on these specific sites. I imagine that will improve the manufacturability in the sense that you can say your population is homogeneous, but then all of these steps add a complexity in the manufacturing process. Making proteins with unnatural amino acids incorporated is not straightforward, and so on, so on. And not cheap either.
Yeah, quite a difficult process to get right, it seems.
Yeah.
Yeah, a lot of moving parts. But does anyone want to talk about any specific deals other than the Myrix deals? Maybe Seagen, Pfizer, pretty big deal that happened previously, but then didn't seem to pan out quite as expected or hoped for thus far.
Yeah, I mean, they've dropped most of that program as far as I understand. So you might be more up in it than I am, but I think the last thing I saw, they've spent quite a lot of money on Seagen, and they're not necessarily taking that forward.
Well, yeah, I believe the assets might have been discontinued, quite a few of them. I'm sure they
Mhm.
have utilized their technology that they had.
Yeah.
So it will be interesting to see what other developments might come from it, as I'm
Mhm.
sure they have quite a lot of capabilities over there. But yeah, it's a tricky field overall. That's just one where, yeah,
Yeah.
that seems to have not as good of a outcome
I mean,
as one might have hoped.
I'm just looking back over my notes on that, and they seem that when they've purchased Siegen, they have taken on a set of assets. I think it's those assets that they're not continuing, but there is a platform there for site-specific conjugation to engineered cysteines. So that is similar to what I was saying before, that this is engineering the antibody to have sites where you're sticking the drug on at a known ratio. So it might be that that looks like a negative signal, but actually this platform is more valuable to them than the very specific assets that they had picked up.
Yeah, I'm not sure if they had some commercial assets as well at Siegen, and that was part of the whole 43 billion acquisition. So I'm sure they've got quite a bit of value from it, but some of the assets, well, it happens no matter what. Some
Absolutely.
I was
assets
just
programs.
reading into some article, and it also stated that on the positive note, they can now use 4.5 billion write-off. So in deal making, it's also about that.
Yeah, it's like in Kathy Woods, like he can use investment in her firms for write-off. Like, thank you very
Exactly.
much.
Yeah, thank you.
Yeah, that's very generous.
I think the Gilead acquisition of Tubulus is quite a prominent one. Again, I think that it's the platform element that is the real moneymaker here. They've got a linker payload combination that is proprietary and is going to impact how payloads can be delivered to cells and can conjugated to antibodies. And I think, maybe I'm looking a bit too broadly, but all these kind of deals are showing me that, there's assets swapping hands. I know you say there might be some already commercial ones, but really it's the linkers, the platforms, the payloads, and going back to Mirix and Novartis, who have just picked up a whole bundle of preclinical payloads with no clinical demonstration of efficacy, but with the understanding that someday you'll be able to stick these onto some sort of antibody. So you don't necessarily need to show that an antibody can do it yet. As long as you've got the asset now, you can tie that on something down the line.
Yeah, it seems to be involving a lot of platform technology in these deals, not just the assets themselves, which, that would make sense why they pay these amounts for some of these.
Yeah. And it also depends on the stage, right? We did see some feedback regarding the earlier, the more likely it's going to be a platform deal. So it also depends on multiple factors, but
yeah, absolutely. Well, I feel like does anyone else have anything more to add before we wrap up?
I mean, there's a lot out there, but I think we've covered
we could talk about, it's these types of payloads. Okay, where do the linkers bind and how likely? Let me see what the question was. There was one specific question.
Are we going to cut this? Because then I'm going to charge my PC. Because it's about to die.
We can cut it. That's fine. I'm going to. Can cut this. Yeah, it's something about. So I didn't discuss this earlier, but let me see. It was during our Pfizer meeting. They asked about manufacturability and stability of the attachment site.
Yeah,
so that's where you talked about how well it actually offloads at the intended site where it's supposed to, and doesn't drop the payload before.
Yeah, yeah. I think that's the
And also,
That's the
they
key thing.
mentioned something typically that they usually have below 3 mg per kg. Typically,
yeah.
overall, it's below 3 mg per kg. They asked about bone marrow tox, since that's a pretty well-known phenomena.
Yeah.
And they also asked about the 2
So
arms.
bone
for the
marrow
linkers.
toxicity. Yeah, is it? So this is questions Pfizer asked you.
Yes.
Yeah. So
I spoke about rabbits. What did I say about the rabbits, Ryan? Pfizer?
I
They asked about the
remember.
rabbits. Remember the rabbits? They asked about the rabbits.
Is that where they're generating the
It's-
antibodies
it's-
from?
It was about
Uh,
Genentech.
No, it was the data for something on rabbits. They mentioned rabbits, but either way. And then bivalent specific, they mentioned something, and then PK data, and then
Yeah.
some number on lung tox.
It was about toxicity of something specific. It was about toxicity.
Yeah,
of
it
something
was
specific,
tox
but I'm-
on something. I don't know if it was for lung. They did it on
Mhm.
rabbits.
They
I
did
think
talk
that
about
might
lung.
have been. Yeah, they asked about lung tox as well, which might be
Lung
good for you
tox.
to know, just in case. In
Yeah.
future, those were some of the questions that Pfizer had.
Yeah.
Most of the
Let me
questions.
take my
The
notes.
manufacturability they asked as well.
Yeah.
Tumor
those were some of the-
heterogeneity.
Yeah.
That was something that they brought up.
The tumor heterogeneity.
Yeah. That's
Tumor
what we touched on.
heterogeneity.
Yeah.
Yeah.
So this were they quite asking questions for Arvik?
Yeah.
Yeah.
Did they also say something about cleavability, cleavable and non-cleavable? Did they say something about
No, they asked about
I remember.
attachments, stability of attachments. They asked.
Mhm.
Yeah, that they did ask about.
So yeah, they're really asking is the drug going to come off in the system?
Yeah,
That's
exactly.
probably boils down to and like what the proportion is. So by the sounds of it, they probably did some tests in rabbits to find out how much of it was coming off. That's probably the. I mean, I don't know. But yeah.
Yeah, rabbits and monkeys.
Yeah.
They did the testing. Yeah.
No llama.
Oh,
No.
no llama this time.
Is that for
Not this time.
nanobodies? Was that nanobody conversations you've seen llamas coming up?
No, but some people they tested on llamas as well. We've seen
China.
certain zeros.
A lot of the
Yeah.
Chinese do it.
Yeah, they do some on llamas as well. We've seen
Mm.
some people ask for llamas for the tox.
Interesting.
Don't ask me why. I'm not a scientist, but apparently
No, I
some
know.
people
It's-
use llama.
I know
I'm
the
gonna
still
llamas.
record.
monkey.
Like, are you?
Yeah, but
Sorry?
I'm gonna cut this anyway in case we
Okay,
add
okay.
I know.
something.
Yeah, I know
Yeah,
llamas are
stop.
big. Yeah, llamas are big in the